In this video, we'll talk about the final part of the immune system, the adaptive immune defenses. The adaptive immune defenses make up the third line of defense, the specific immune response. If you remember, previously we talked about the innate division, which has the first two responses, the physical and chemical barriers, and the non-specific internal immune responses, which we discussed. Now here, we're focusing on, like I said, adaptive immunity. This is a little bit slower, at least for the initial response, but it's highly specific. So we call this specific immunity. There are three main groups here you want to think about: T cells and B cells, and these proteins called antibodies that are actually produced by a specific type of B cell. So these two main cell types, T cells and B cells, make up this adaptive division. So you can see over here, both of these cells, like all blood cells or all formed elements, start out inside our bone marrow as stem cells. In this case, some go on to become T cells, which will migrate to an organ called the thymus, where they mature. So T is short for thymus. And then the B cells will migrate to a different part inside the bone. They'll stay there, called the bursa. Anyway, B for bone to think easier. They stay there, and they have different roles. The T cells are involved in what's called cell-mediated immunity, meaning that they're directly involved in attacking cells. T cells are attacking infected cells, our own cells that have been infected. Whereas B cells are involved in antibody-based immunity, also called humoral immunity. This is where they produce these proteins called antibodies that then help to destroy or mark things for destruction that are outside of cells. So B cells aren't doing direct attacking, but they're producing antibodies that attack things that are outside of our cells, or mark our own cells for destruction by these T cells. And we'll get more into each of these one at a time. So you have a certain stockpile, for lack of a better word, of white blood cells. But what happens is when they get stimulated, when they are presented with antigen, that stimulates them to divide, to begin building this clone army. This is called clonal selection. So an antigen is presented to these T cells or B cells. Lymphocyte is another name for T cells and B cells. We will not use that term in this class, but if you go on to anatomy or microbiology, you may use that term. So T cells and B cells are stimulated to build a clone army of identical individuals to go and recognize and fight off that very specific invading pathogen. From those clones, there are two different types of cells that are made. One of them is a category called effector cells, with an E. These have a direct effect, direct involvement in the current process to help destroy pathogens or to inactivate those antigen-containing substances. And then the other category are memory cells. Memory cells are not involved in the current immune response. They last a long time, however. In some cases, they'll last for your entire life. And they stick around so that if you encounter the exact same thing in the future, the same antigen, now the memory cells will divide and produce a whole bunch of effector cells to now help fight off that pathogen very quickly in the future. And more memory cells will be made to, again, have that lasting protection. So memory cells are what provides you with lasting immunity. If you don't have memory cells, you do not have lasting immunity. So let's break down the effector cells, talking about the T cells first. So you have these three main types of effector T cells. Helper T cell, a cytotoxic T cell, and a regulatory T cell. Let's talk about what each of these does. So first, a helper T cell. As the name suggests, it helps other cells. It helps to activate them. It's one of the main activators. Helper T cells will stimulate the production of other immune system cells to help fight off invaders. So in this case here, a helper T cell is responding and causing these cytotoxic T cells and these B cells to get activated, leading then to their direct action to help fight off the current infection. So they help to stimulate other immune system cells that are part of this adaptive division. Cytotoxic. Cyto means cell, so cell toxic, also called killer T cells. Similar to natural killer cells, which were part of the second line of defense, the non-specific defenses, part of the innate division, here the cytotoxic T cells, part of the adaptive division, they fight off the same stuff. They target our own cells that have been infected with a virus and cancer cells. If they're abnormal, they will destroy them. In rare cases, some bacteria do go inside our cells and infect us, like is the case with tuberculosis, but usually bacteria stay outside, so we won't worry about that for this class. So cytotoxic T cells destroy our own cells that have been infected with a virus or have become cancerous, and they'll destroy them. And lastly, for the effector T cells, the regulatory T cells. This regulates your immune response after the infection is gone. So once you've successfully fought off that pathogen, you want to slow down and reduce the immune response. One, because it takes a lot of energy. Your immune system uses something like 1/3 more energy than you do when you're not sick. That's why you're so tired all the time. And also, if your immune system is overactive but there's nothing to fight off, sometimes you can start to attack your own healthy cells, right? That's autoimmunity. Or you can attack things that are harmless. That's allergies, where you're attacking something that's not a threat, but your immune system makes a mistake and thinks it is. So the regulatory T cells are key for keeping your immune system properly functioning and stopping or that major defense once it's done. Now moving into the effector B cells. These are called plasma cells. I briefly mentioned these before, but plasma cells are a type of B cell that produces and secretes these proteins called antibodies. It releases them into the plasma, the liquid portion of the blood, where they will attach to a very specific foreign antigen target. So each antibody only reacts perfectly with one specific type of antigen. Our body can literally make millions, even possibly billions of different kinds of antibodies to react to almost all the different types of antigens you can imagine. Now, it's important to consider that one pathogen sometimes has multiple different types of antigens, and so that means that you'd have different antibodies produced against each of these different antigens. So what do antibodies do? Well, they do a lot of things. We'll focus on some of the major functions here. And if you want to on your own time, you can take a look at this video. Again, use your phone, your camera, and scan this little QR code down in the corner. But antibodies will attach, like I said, to foreign antigens. And if it cross-links or connects like handcuffs two pieces of antigen on two different pathogens, it can cause them all to get stuck together. That makes it easier then for the body to prevent them from spreading, kind of holds them all together in one group. And it makes it easier for phagocytes to do their job of going around and gobbling up this group of pathogens instead of chasing after each one at a time. It makes it more efficient. Remember that antibodies and previously in the innate division we talked about complement protein. Both of these make it easier for phagocytes to find their target and engulf it through phagocytosis. So they both enhance that process. Now let's talk about antibody-based immunity in a little bit more detail. So you can have active immunity versus passive. Active means that your immune system is actively being stimulated, which leads to the production of antibodies by your own body. Passive means your immune system is not stimulated. Instead, you're being provided antibodies that were made by somebody else or something else. So active, immune system stimulated, you make your own antibodies. Passive, you're being given antibodies from some other source. Now let's take a look at natural versus artificial for the active immune division. Natural means you encounter a pathogen out in the world. You get sick. You catch a cold. You get chickenpox. That provides you with natural exposure, which stimulates your immune system, active immunity. You produce antibodies and memory cells that will protect you. Artificial active immunity means it's something that we've done. We as humans inject pieces of a dead virus, for example, into our body as a vaccine to stimulate our immune defenses to fight against something that's not in a harmful state, but where the real thing could hurt us. So it gives us the same kind of stimulation or similar that an actual infection would, but without the threat and the danger. So when I was young, for example, I got chickenpox. I actually caught that infection. I have natural active immunity that developed from that. Some of you may have gotten the chickenpox vaccine. You have artificial active immunity. Not necessarily one better than the other. Sometimes not all vaccines are 100% effective, but both of them give you lasting protection via the production of memory cells. Now over here, natural passive immunity. One example of this is when a woman's pregnant, some of the antibodies she's producing cross the placenta. We'll talk about that more later, but it's a protective organ that surrounds the fetus. And they go in and they help to protect the fetus from infection. Or when a mother breastfeeds, she will then transfer antibodies through her breast milk to help that newborn fight off infection until it can really ramp up its own immune defenses. But that's temporary. Antibodies being given. It's not stimulating the immune system, so it's passive. Similarly, if you get an injection of antibodies, let's say you step on a rusty nail, or you get bitten by a dog and they think it might have rabies, you get an injection of antibodies against the rabies pathogen. In that case, that's artificial because we're injecting something or putting a man-made thing into you, and it's not stimulating your immune system. It's giving you a temporary boost, but not lasting protection. So the only way I've said before you get this lasting immunity is with memory cells. They can live for decades, in some cases for your entire life. So you have different kinds of memory cells. You have memory B cells and memory T cells. Once you encounter that same antigen again, they'll produce effector B and T cells to go out and help fight off the current infection, and more memory cells to stick around again for later. So we said that the adaptive response is a little bit slower initially. So over here is your very first time you've been exposed to a particular antigen. Your primary immune response by the adaptive division is relatively slow, and antibody levels over here, the concentration, doesn't get very, very high. But now you encounter it again later on in life, maybe even a short while later. The immune response is very rapid due to having those memory cells. You have a very quick response to the formation of those effector cells. It's also much stronger. You can see that the antibody levels are vastly higher. And so when we say you're immune to something, it doesn't mean the pathogen can't get into your body. It can. But what happens is your adaptive division wipes it out so quickly, you don't even know that it got in there. So it's a very rapid and strong secondary response, secondary immune response. And those memory cells then are going to help with that process. So the general steps here: recognize, respond, remember. So an infection is recognized, stimulation happens, that leads to clonal selection. Clone armies are produced, creating effector cells that go off and are involved in the current infection. Those are the different B and T cells. And then memory cells that last, like I said, for long term. Here's a cartoon to kind of summarize this. I think it does a great job of simplifying it and making it very digestible. So here we have a dendritic cell. We didn't talk about this, but this is a type of antigen-presenting cell, which we did talk about. It's a special type of phagocyte that gobbles up pathogens and keeps a part of it as a trophy, a little piece of antigen. and brings it, in this case, to a helper T cell. It shows the helper T cell, hey, look what I found. The helper T cell gets excited, it gets activated. The helper T cell then does its job, which is to go help activate other cells. So here we have a B cell that's encountered some bacteria, and one of them fits. And like, I think I should make antibodies against that, but I won't do it without confirmation. So the adaptive division always waits for this confirmation from the helper T cell. And remember, the helper T cell had just been activated by that antigen presenting cell. So it's like, yes, helper T cell, yes, B cell, this is something we should fight off. So now the B cell gets activated and it produces the plasma cells. Those are the effector cells. And then the memory cells that are involved later on. Plasma cells make a whole bunch of antibodies, those proteins that are going to attach to these foreign antigens present in our body, helping to mark it for destruction by phagocytes that'll come and they'll gobble it up and destroy it. And memory cells last then long term. Here's a summary slide showing this whole branching of the two divisions with the summaries of each. I'm not going to go through this, but it's a good summary slide to kind of look back at as you're studying. Here's another one, looking at the regulatory T cells, how they'll slow down that response after the infection has been cleared. And then I think this is close to the last slide here, comparing and contrasting innate versus adaptive immunity. So innate, remember, was the first and second lines of defense, and adaptive was the third line of defense. Innate is immediate, it's inborn, ready to go from the very beginning. Adaptive takes some time for the first primary exposure. In the future, it's much faster. That can take a week or even 2 weeks potentially to get the initial adaptive response up and running. How specific are they? Well, remember, innate's a very general, non-specific defender. For example, natural killer cells would target the same type of marker present on all viruses or present on all cancer cells, whereas the adaptive is extremely specific. Only one type of antibody is detecting one specific type of antigen. Persistence, how long does it last? Well, innate's always there, but there's no lasting memory. Unlike adaptive, if you have those memory cells formed, you have now lasting protection, lasting immunity provided by that adaptive defense.